Visualizing a single-cell sequencing dataset can be a very challenging task since there are multiple layers of annotations on top of the expression matrix. On BBrowserX, we provide a simple interface to visualize and interact with metadata. Now, let's learn how to navigate the BBrowserX interface and manage metadata. When you first enter a study, there will be a scatter plot of all the cells in the dataset in the center of the interface, the metadata panel on the right-hand side, a toolbar at the top, a sidebar on the left-hand side, and a section for gene query and visualizing gene expression data. With the scatter plot, you can zoom in, zoom out, reset the zoom level back to the default, use the lasso tool to select cells, or change the point size to have a better look. You can also export the scatter to Vinci for editing. The cells on the scatter are colored by the groups in the metadata section on the right-hand side. A study from BioTuring's database contains BioTuring's standardized metadata and all authors' metadata. Your annotation, including imported annotation and manually labeled metadata, will be added under your metadata. For categorical metadata, different groups of the metadata field and their respective number of cells are shown under the drop-down menu and are visualized with different colors on the scatter plot. Check or uncheck the box next to a group name to show or hide a particular group on the scatter plot. For numerical metadata, a distribution plot and a box plot are shown. To change the color palette, click the Edit button, then switch to the Color Palette tab. You can change the color palette for categorical metadata, numerical metadata, and expression value. To manually annotate a group of cells, first, select cells by choosing a group in the metadata panel. Press and hold the Control or Command key to select multiple groups. Using mouse clicks on the scatter plot. Using the lasso tool to locate cells. Or selecting a range on the legend. The number of selected cells is shown on the top of the scatter plot. Then, click the Label button. In the first input, fill in the field name to store the label. Select the Create option to create a new field. You can also append the cells to an existing field, which matches your input. In the second input, enter the name of the cell group. Similarly, you can create a new group name or append to an existing one. Another way to add metadata is to upload a metadata file. Click on the Edit button, then choose Import from File. Make sure that your file consists of the exact barcodes of each cell and its labels. Different metadata fields can also be combined to create new fields. In the Edit window, fill in the name of the new field and select up to 3 different fields to generate all the combinations of the fields you select. You can click on this button to edit or delete the currently selected metadata. To make changes or remove multiple metadata fields at once, click the Edit button, then switch to the Rename or Delete tab. And finally, you can export all of your annotation into a TSV file or along with the expression matrix in a Scampi or Seurat object. We hope you find this video helpful. If you have any questions, contact us anytime via support@bioturing.com.